Hello, everyone. Welcome back. It is now my honor to introduce Patrizia Cavazzoni, who is the director of the Center for Drug Evaluation and Research, CDER, at the FDA. Patrizia, who is certified by the American Board of Neurology and Psychiatry, joined the FDA in 2018 as CDER's Deputy Director for Operations, where she has led several key initiatives on behalf of the FDA. She was appointed director of CDER about a year ago, and I know that everyone here knows that Patrizia had to hit the ground running with the pandemic and the emergency use authorizations, et cetera. And she has done a fabulous job. Please join me in welcoming Patrizia Cavazzoni, who will speak with us about expedited regulatory pathways and inclusive clinical trials.
Hello, everyone. I'm going to share my slides and get started. All right, very well. So I'm very pleased to be joining today, and I'm delighted to be walking you through a couple of topics that are really very relevant to meeting unmet needs in both rare diseases and higher prevalences diseases. So I'm going to touch on expedited regulatory pathway with a focus on accelerated approval. And then I will also briefly touch on the importance of inclusiveness and diversity in clinical trials as a way to address unmet needs in both rare and higher prevalences diseases. So while rare diseases certainly get a lot of attention when it comes to unmet need, let's not forget that there are a number of higher prevalences diseases that might have standard of care, but also present a significant residual unmet needs, such as treatment-resistant depression, for instance, PTSD, Alzheimer's disease, and so on. So when we look at rare diseases as well as more common diseases that still have residual unmet needs, we can see some commonalities, including the fact that the regulatory pathway may not be well defined. Also, both may require unique programs and study designs, and most importantly, both may benefit from early interaction with FDA to support efficient development. And we actually, as part of the PDUFA 7 proposals, there is a new meeting prior to IND to address unique development challenge and start an earlier dialogue with sponsors. There's a number of challenges in rare disease development. I will mention just a few. The natural history of diseases are often poorly understood. The diseases tend to be progressive, life-threatening, and lack approved therapies. The populations are small. The drug development tools are often lacking, and there's also very often a lack of precedent for drug development. When it comes to the higher prevalence diseases, they also present a host of challenges, such as the challenge with identifying the subpopulations where there may still be residual unmet medical needs, such as resistance disease, such as patients who remain inadequately controlled, for instance, in type 2 diabetes, diseases with new options, but residual substantial unmet needs, such as obesity, or diseases where, despite incredible advances in many therapies, we're still seeing a lot of residual mortality, such as coronary artery disease. Additional unique challenges to higher prevalences diseases are clinical trial recruitment, particularly when it comes to identifying subsets where there may be higher unmet need. Trial designs may require superiority to standard of care or add-on to a standard of care, and identification of treatment-resistant patients may be challenging. In these diseases, there's also a need to consider flexible and innovative approaches. There are a range of less common designs may have to be considered, such as finding treatment, randomized withdrawal, superiority to standard of care, and so on. Master protocols and platform trials to study a range of treatments at once, and also limiting the exposure of placebo or what might be, at least presumptively, less effective standard of care. Adaptive design trials to improve trial efficiency are also an important tool, and also novel endpoint development, which may enhance trial efficiency. There are sorry for doing that. There are drug development in areas with few or no existing therapies pose unique challenges. Often we see limited programmatic experience from sponsor staff. There may also be limited investigational site experience, which may lead to recruitment or retention challenges in clinical trials. There may also be lack of prior precedent for utilization of a regulatory pathway or expedited pathways, and we may not have guidance that has been issued by FDA because of the novelty of either the disease state, the subpopulation, or the characteristics of the development programs. And there may or may not be existing endpoints with well-established characteristics. Now, FDA has a host of expedited pathways that are available to us by statute. These include fast track, breakthrough designation, priority review, and accelerated approval. And I'm going to be focusing on accelerated approval from this point onward. So there are two pathways for approval, both based on adequate and well-controlled trials. The first is traditional approval. The second is accelerated approval. In both instances, the approval is informed by several factors. The extent of the effectiveness, meaning what we see when it comes to the symptomatic benefits versus mortality, for instance, and where we see the effectiveness. The safety profile and the risk to patients, which has to be balanced against effectiveness, understanding of what matters to patients, including the value of potential benefit of the drug, the tolerance for risk by patients, and also determining the level of uncertainty that is acceptable about benefit or risk depending on the severity of disease and the degree of unmet need. When it comes to traditional approval, it is based on a clinical endpoint that reflects how a patient feels, functions, or survives, or a validated surrogate endpoint. And I think I really want to emphasize that the validated aspect of this, because I will come back to this when I talk about accelerated approval. A validated surrogate endpoint is an endpoint that is a surrogate that is known to predict clinical benefits. For instance, systolic blood pressure for cardiovascular events or hemoglobin A1C for diabetes. The efficacy, when it comes to traditional approval, the efficacy is established in registrational trials. So additional studies are not required in the post-market setting to confirm the benefit, although very often we require additional safety data when we think that the disease state or what the data in the program warranted. Now, let's talk about clinical endpoints, and this pertains to both pathways. Some diseases progress very slowly or have a variable course in patients, and neurological and neurodegenerative diseases such as Alzheimer's or ALS are good examples of this. And trials based on clinical endpoints for these types of diseases may require, therefore, large sample sizes, which may not be feasible for uncommon diseases, and long study durations, which may not be practical. Now let's switch to accelerated approval. This is at a glance the short history of accelerated approval. It all started with the HIV crisis in 1987. AZT was approved based on a single placebo-controlled clinical trial and this generated a lot of controversy at that time because up until then the true controlled clinical trials sort of paradigm was the norm, and so this was rather unprecedented. In 1992, Congress enacted a statute that gave FDA the authority to approve based on a surrogate endpoint that is reasonable, likely to predict clinical benefits, and shortly afterwards, accelerated approval was deployed to approve additional antivirals for HIV. And since 1992, as of June 2021, we have approved 260 drugs using accelerated approval. Now, when it comes to accelerated approval, there are two very important elements that I really want to highlight today. So this comes straight out of the statute. The basis for accelerated approval has to be based on an effect on a surrogate endpoint that is reasonably likely to predict clinical benefits. And this is different from known to predict, which we expect in a validated endpoint, which can serve as the basis for traditional approvals, or on a clinical endpoint that may be measured earlier than irreversible morbidity or mortality, which may be upstream in the disease course, but is also reasonably likely to predict an effect on morbidity or mortality. And also by statute, we have to take into account the severity, the rarity, the prevalence of the condition, and the availability of other therapies when we are considering accelerated approval. So let's talk about this concept of a surrogate that is reasonably likely to predict benefit. Such a surrogate would entail a marker such as a laboratory measurement, a radiographic image, a physical sign, or other measure that is not itself a direct measurement of clinical benefit, but that is reasonably likely to predict clinical benefit. This requires data to support that the surrogate predicts a clinical benefit of interest, but allows for more uncertainty that would be expected for a validated surrogate endpoint. And this concept of a residual uncertainty is fundamental in thinking about accelerated approval versus traditional approval. We require post-marketing confirmatory trials to verify and describe the anticipated clinical benefit, and FDA has the statutory authority to withdraw approval if the trial fails to verify the clinical benefit, or if does not demonstrate sufficient clinical benefit to justify the associated risk. So by design, accelerated approval tolerates greater uncertainty about benefit and risk at the time of the initial approval decision compared with traditional approval. And this is really fundamental in the concept of addressing urgent public health needs and very serious, significant unmet medical needs faster than traditional approval would allow. The standard for reasonably likely to predict clinical benefit should be set at such that clinical benefit is subsequently verified in the post-marketing setting for a high proportion of approvals, but not all. And in fact, we have had a number of instances where the confirmatory trials did not confirm efficacy after the drug is approved through accelerated approval. And in fact, this is something that we should expect because if we had a situation where the benefit is confirmed for every single drug that is approved through accelerated approval, the threshold may actually not be set to appropriately balance the early access versus the residual uncertainty that we might consider acceptable within the context of a situation where there is a unmet medical need. Now, what are the challenges with accelerated approval? The surrogate endpoints can improve feasibility, but the challenge in many diseases, including in particular many neurological diseases, is that there is very limited understanding of the pathogenesis of the disease. The data to support the relationship between the surrogate endpoint and the clinical benefits are often limited, and as well as information that is critical to establishing a biomarker as a surrogate endpoint. And again, I come back to neuroscience and neurodegenerative diseases. This is pretty much the norm, or has been the norm in neurology, which is why there haven't been many accelerated approvals in neuroscience. And also, many animal models of disease do not fully characterize the key aspects of the human disease and are not translational. So at a glance, if we see the relative proportion of accelerated approval, we clearly see that oncology is where accelerated approval has been used the most, followed by hematology, and then in other areas, infectious diseases really represents the bulk of accelerated approval. Why has oncology, why have we been able to use accelerated approval in oncology to a much greater extent compared to other therapeutic areas? Is because the key driver in oncology has been a huge advances in the understanding of genetic, molecular, and immunomodulatory drivers of cancers in the 90s and 2000s, which then sort of translated into this peak of accelerated approvals later starting in 2010. And this is an important, an essential characteristics that really explains why we have been able to use the accelerated approval in oncology to such an extent. Now, although we have used accelerated approval in oncology to an infectious disease to a much greater extent than in other therapeutic areas, it can and should be applied to other disease areas as the scientific evidence evolves. And this improved scientific understanding of both the disease and the role of specific biomarkers may lead to the availability of surrogate endpoints in the future that could support approvals in other disease areas, including in neurology. And we have seen an example this last year where we were able to utilize the amyloid, the reduction in amyloid, to support approval of a drug for Alzheimer's. Having established that reduction of amyloid reasonably predicted clinical benefit in Alzheimer's disease. I'm going to say a few words about inclusion and diversity in clinical trials. I would like to spend a lot more time on this because this is so important. But inclusion and diversity in clinical trials is essential to, number one, making sure that clinical trials are generalizable to the populations that will be taking the drug after it is approved, but also to accelerate development and making sure that we meet unmet medical needs. And that means also making sure that we meet unmet medical needs in all the populations that are going to be taking the drug once it's approved. Earlier in development, very often we see homogeneous populations. But however, it is really very critically important that the populations be broadened later in development in phase 3. And it is very important to consider demographic characteristics and the differences in also background diseases and disease morbidity in various populations as enrollment in clinical trials is planned. Now, this is an area of great interest to us. We have issued guidance to guide sponsors on how to maximize diversity in clinical trials and enrollment of underrepresented populations. We're going to continue to do some work in this area, and we think that this is an area that remains is extremely important, and we need to do better. As one can see in the clinical trial, in the drug trial snapshot that we have posted on our website, we're doing much better in certain therapeutic areas, but there's a ways to go in other therapeutic areas. And I gave the example, for instance, of Alzheimer's trials, where I think there is a lot of room for improvement when it comes to the enrollment of more diverse populations. Technology is also very important in ensuring that recruitment reflects the population for which the drug is ultimately designed, and also to make sure that underserved populations are given the opportunity to participate in clinical trials in disease areas where often the clinical trials may actually be the only therapy that is available for a specific condition. And so, as we have seen during the pandemic, we have been, by necessity, we have had to really expand the use of decentralized clinical trials, digital health technologies, drug development tools, and so on. And this has really yielded some really great successes during the pandemic, because we have been able to continue to advance clinical trials during times where patients really could not make it to physical sites for long periods of time. And certainly, at least at a minimum, these tools limit the need for patients to have to go to the sites for every single activity or in the clinical trials. So I'm going to sort of highlight a couple of things here. The couple of things that I really wanted to sort of flag is that it is extremely important that we leverage accelerated, expedited pathways to address these unmet medical needs, both in rare diseases and high prevalence diseases, and that we really have to leverage, technology and flexibility when it comes to how clinical trials are executed to make them more inclusive and more representative of the general population. I'll stop there and turn over for maybe we have time for one question.
Thank you, Patrizia. Thank you very much. That was a great presentation, and we learned a lot. We do have a number of questions, so I'll pick a couple of them. So one of them is, given that natural history is poorly understood and hard to obtain in rare diseases, is there an opportunity for FDA to encourage data transparency and public disclosure of the control arm data from completed studies?
Yes, absolutely. And in fact, we do have initiatives in rare diseases that are really pointed to that, to facilitating the understanding of rare diseases and to get to greater data sharing. I mean, the Rare Disease Accelerator is really the emblematic of the effort in that space. So it is really essential that we try to really make the study of the natural history of diseases more efficient for rare diseases. And also these placebo, sharing of placebo data would also help us in situations where we could use, for instance, external controls that might then minimize the exposure of patients to placebo.
Yeah, I think that'd be a great idea. Let me ask you, there's a lot of talk about using real-world data, of course, but there's also people at the front end using model-informed drug development. So how does model-informed drug development, how is that used, to, either replaced, either for rare diseases where they put computational models? How does FDA consider that?
Well, drug and disease models are relevant and useful for both rare and high prevalence diseases, and in particular when it comes to high prevalence diseases, for when we're trying to look at subpopulations, and the use has to be expanded. We have a model-informed drug development program at CEDAR, and it has expanded significantly. And I think it's the way of the future. The future is moving more towards a totality of evidence, a paradigm when we look at that data, and simply because we have to start thinking about how to bring in, how to consider real-world evidence, how to consider disease and drug and disease modeling, and so on. And so modeling is going to become lead from be a leading science from a quantitative standpoint.
Yes, I agree. Thank you. Thank you, Patrizia.